Hi guys, welcome to Let's Get to the Marks. We're looking at the methylation of DNA. So we've got two strands here, the double helix, one strand in pink, one strand in green. And I'm now going to draw some bases. I'm only going to draw 4. I think 4 will be enough. Normally a gene has thousands of bases. So a short section of DNA is a gene. And I'm really interested in 2 of these bases. And it's when cytosine and guanine are next to each other on the same strand. This is called a CpG site. And the C stands for cytosine. The P stands for phosphodiester bond. And the G stands for guanine. Oh, I've typed phosphodiester bong. Well, we don't want to do one of those. That sounds pretty dangerous. So yeah, cytosine, phosphodiester bond, and guanine. Now the CpG site is going to have a different 3-dimensional shape than any 2 other adjacent bases. So it will be a different shape than the GA site. And the CpG site has a complementary shape to a methyl group, which is a carbon with 3 hydrogens attached. And that means that the methyl group can enter into the DNA molecule and it can attach to the CpG site, just like I've shown here. And what does this mean? What is the consequences? Well, RNA polymerase, should it want to bind to the start of a gene, which has a CpG site, it's going to have trouble. It's not going to be able to access the bases where it can attach. And this is going to affect transcription. In fact, this is going to stop RNA polymerase from transcribing that gene with the CpG site. But what else cannot access the gene? So we have to think, what is the other type of machinery needed for transcription? And often you have transcription factors, and you might remember there are repressors and activators. Activators are things that stimulate transcription and therefore increase the rate of transcription, things like estrogen. The result of the methylation of the CpG sites is that these transcription factors cannot bind to the DNA where they normally aid the binding of RNA polymerase. And the overall result is that the gene isn't transcribed, so we don't get mRNA for that particular gene. Effectively, the gene is switched off. So we say that methylation of a gene means it can be switched off. And what are the consequences of that? If you don't produce mRNA, then you don't get translation on the ribosome and you don't get the protein produced. Now let's imagine that that protein that we want to produce from this gene here is one that suppresses tumor production. It prevents the production of a tumor. Well, you can see how methylation of this gene could end up with the formation of a tumor. Okay, guys, that's us done really, just circling the transcription machinery here. And that's methylation of DNA and how it can lead to tumor production and how it can switch genes off. All right, thanks. Keep watching. Let's get to the marks.